Hello everybody, my name is Anel and we are going to talk about hemidesmosomes. They are kind of similar to desmosomes, especially when you look at them through a microscope, but they are different in structure and kind of in function, so we are going to talk about both of them. They might also be similar to focal adhesions, so I'm also going to explain them, but quicker, because I have already made other videos about them. So, the basic idea of hemidesmosomes is that they connect the intermediate filaments, that is, one kind of filament in the cytoskeleton. The thinnest one is microfilaments, the thickest one is microtubules, and in the middle of those two you find intermediate filaments. They are intermediate, but they are also the most resistant, so don't be fooled by that. Hemidesmosomes connect intermediate filaments to the basal lamina. That is one kind of extracellular matrix, the one that you often find below epithelia, because hemidesmosomes you find them in epithelia. Epithelia is the kind of tissue that surrounds our body or lines the parts of our body that are exposed to the exterior. Our skin, the mucosa that covers our mouth, our airways, our digestive tract, all of that. It's oftentimes polarized, with one side that might have some special features, like cilia in our airways, the microvilli in our intestine, and another side that is looking to the basal lamina, and which will have hemidesmosomes. Hemidesmosomes don't connect one cell to another cell, they connect one cell to the basal lamina. And basal lamina is one kind of extracellular matrix. Extracellular matrix is a combination of proteins and glycans that basically gives shape to tissues or to organs and helps cells to stay in the place that they must stay. And the main proteins they use to do that are integrins. Desmosomes, on the other hand, connect one cell to another cell. They connect the intermediate filaments of one cell to the intermediate filaments of another cell, a neighboring cell. And they don't use integrins, they use catenanes, a different kind of transmembrane protein. But because both of them are linked to the intermediate filaments, both are involved in the same kind of overall function, which is to resist mechanical forces. If you think about it, there's a lot of friction going on around our body, by our clothes or when we grab things, and inside it, in our mouth, in our intestines. If those cells didn't have strong attachments to their surroundings, or their cells, or the extracellular matrix, they would be pulled off, and that would be pretty dangerous because it would expose the underlying tissue, making it prone to infection or to receiving other kinds of damage. So in the end, hemidesmosomes and desmosomes both are trying to protect cells against friction or other mechanical forces by attaching them strongly to other cells or to the extracellular matrix through the intermediate filaments, but they do it in different ways. Focal adhesions, on the other hand, connect to the microfilaments, which are not that strong, and although it helps cells to migrate, to test the properties of the tissue that surrounds them, it's not so good at resisting forces, so the function is different in that case. But they also use integrins in a similar way as hemidesmosomes did. Alright, enough of that. Let's talk about how to make an hemidesmosome, what proteins it's made of, because we have already mentioned some of them, but not all of them. The basic elements we need are intermediate filaments, a membrane, the membrane of the cell, in the basal side to be more precise, and the extracellular matrix, or in this kind, the basal lamina. The main proteins in the basal lamina, by the way, are collagen, laminins, perlecan, and nidogen, or nidogen. I don't know how to pronounce that actually. I'm rushing through them because I will make another video about the basal lamina, and now it's not the time to start talking about all those proteins and how they interact with one another. We also need transmembrane proteins. The main ones, as we said, are integrins. Integrins have two subunits, the alpha subunit and the beta subunit. The most common kind of integrin in hemidesmosomes are alpha-6 beta-4 integrins. But integrins can't interact directly with intermediate filaments. They need adapters. Oh, you know this. The main adapter for integrins in hemidesmosomes is plectin. And with this, we have the main idea, or the main model of hemidesmosomes, the simplest one too. We can go a bit further. Another transmembrane protein that we could find in hemidesmosomes is what we call BP180 or BP180. BP180 also needs an adapter, BP230. By the way, BP230 can also interact with the beta subunit of integrins, so those tend to cluster as well, as you see in this drawing. Of course, in a hemidesmosome, you have hundreds of these things. But that's it, we don't have secondary adapters or other adapters and all of the complex things that we had in other kinds of junctions. Hemidesmosomes in that respect are pretty simple. Here are a couple of fixtures to summarize everything. I would recommend using Anki to remember these things. And you can download the slides in the channel's website, flipyourlearning.com. I'll leave you a specific link for the page where you find the slides for this video in the description below. The next video is going to be about focal adhesions, and let's be honest, those are complicated. They have a lot of adapters and different ways of connecting integrins to the microfilaments, and they also have a lot of signaling involved, and regulation on how you create them, how you make them grow bigger, and how you make them disappear when you don't need them anymore. Because hemidesmosomes you probably need all the time, but focal adhesions not so much. Alright, I hope to see you soon. Bye, hasta pronto.